Okay, well, hello everyone. Now that we're being recorded, welcome to the fall semester of 2020. It's definitely going to be one to remember. And to inaugurate our seminar series, which is now almost interplanetary, I guess would be possible with the new remote format, we decided that the best person to start the seminar series would be someone who is extremely well versed in soft matter physics and all the stuff that we usually talk about, but is also one of the, I would say, pioneers in doing real biophysics, meaning that Margaret Gardel is really one of my heroes because she is a woman who is not afraid to go and work with real cells and talk to real biologists and ask real questions about how life began or goes on. And so, as far as I can tell from the abstract, she's going to be telling us about design principles, which is very soft mattery material science. She's also, by the way, the director of the MRSEC at Chicago. So there's a bit of healthy competition going on there, I hope. Well, it used to be anyway. And so she will tell us about soft matter aspects, I assume, or material science aspects to how living matter emerges and evolves into organisms through morphogenetic principles of design. So welcome, Margaret.
Cool. Thank you very much for having me. I'm excited to be attempting to give this talk. And we'll see how it goes. Please, if you have questions, use the chat because I can't get any other types of cues about whether what I'm saying is comprehensible or not. So as Yasna said, my lab studies questions about how cells and tissues form and change their shape. This I saw as an opportunity to recap some of the soft matter work that my lab had done over the last several years, and I realized I hadn't organized a talk on this, so if it's a little bit discombobulated, I apologize for that. So we're really interested and motivated by really thinking about cell motion and cell and tissue morphogenesis. So these are two just phase contrast movies that you see at the scale of individual cells and at the scale of a whole embryo of the shape changes that the materials within cells drive as they're undergoing motility through a matrix. The top movie is that of an endothelial cell from a blood vessel wall migrating through a collagen network. And you can see it's generating forces on this network. It's protruding out in the front, detaching from the rear. And the rightmost movie is the first 24 hours of a zebrafish's life as it goes from a single cell to a very complex multicellular organism with structure. So I'd like to sort of summarize the last 100 years of very successful molecular biology in terms of identifying the molecular scale, at the molecular scale and the genetic scale, how the basis of these morphogenic properties. But even though there's been significant advances in that, and that has enabled sort of the engineering of genetic materials of cells, it still leaves us with this question of this vast span of length scales going from the molecular all the way up to the cellular scale of tens of microns of how do you build materials out of these molecules that enable these types of shape changes and sort of mechanical robustness you see at the cell and tissue level. So my lab has studied for the past 12 years, how do you describe the mechanics of the materials within the cell? For the types of cells we study mostly epithelial tissue. The actin cytoskeleton, which I'll go into more details in the next few slides, is really the important mechanical framework of cells. And it's the actin cytoskeleton that allows cells to generate contractile forces, generate protrusive pushing forces, modulate the adhesion to the extracellular matrix, the external environment, or between cells, and also just modulate and maintain the cell shape, the actin cortex. So this is a dynamic force generating and sort of adaptive polymer network that is really essential for cell morphogenesis. So the way we're thinking about characterizing the actin cytoskeleton now is, and I'm going to only talk about this first one, this force generation aspect, is we are interested in problems both in how in this mechanical network, mechanical forces are generated within this soft material, but also how they're sensed both at the molecular scale, but also at sort of the micrometer scale, and how these two properties couple together to build materials that can spontaneously control, generate forces, control where and when these forces are generated, and respond to external forces. So this is sort of the toolbox in which you build active, so force generating, and also adaptive materials. And the way my lab studies this is sort of a twofold, a bottom up and a top down approach. Sort of the soft matter side of my lab reconstitutes soft materials made out of actin filaments with other types of adapter and binding proteins, and reconstitute morphogenic processes that you might see that are reminiscent of those in cells. But also we study morphogenic properties in epithelial cells and tissues and sort of control those to understand the biophysical regulation of cell shape of living cells. So today I'm really going to talk about the reconstitution, and for the students in the audience, go through sort of the history of how do you characterize these soft polymer materials. So to study these actin-based materials outside the cell, you need to start with the protein and the polymer based on the protein. So the actin polymer itself you form from grinding up muscle, say a rabbit or chicken. You use biochemical methods to solubilize and separate the globular proteins. And in the presence of ATP and a divalent salt, these spontaneously polymerize into polymers. The polymers of actin are polar. They have a distinct polarity at their ends. So there's one called a barred end, where there's a higher on rate of monomers. And then there's a pointed end where there's a higher off rate of monomers. So there's a dynamic steady state of exchange of monomers with the polymer pool. And so this, it's a helical polymer. So it has a significant rigidity when you compare it to, say, a standard polymer you would learn about in a polymer science class, like polyethylene. Right, and the way we characterize the rigidity is you look at the ratio of the bending modulus of the polymer to thermal energy. This gives you a length scale. This length scale for actin polymers is 17 micrometers. So if you polymerize an actin filament and look at it under high resolution light microscopy, so this is a fluorescently labeled actin polymer, and you can see that it's straight, but it has some bending undulations. So this semi-flexibility gives rise to really interesting polymer science questions, because I'm unmuted now, right? Okay, good. It's somewhere between a completely rigid rod where you don't have to think about entropic effects and a completely flexible polymer where all you think about is entropic effects. Okay, so if you, people have measured the force extension of this polymer. This is a summary from a review article that a student of mine wrote, where you look at the force as a function of the end-to-end distance of the polymer, and you imagine applying an extension force or a compression force to this polymer. And so you can see different regimes of the force extension. So the black line is the complete force extension. And in dashed red is what you would expect just for a linear entropic spring of the same length. And then the blue is what you'd expect just for an enthalpic for a rigid rod. And so you can see the semi-flexibility where the curve deviates, sort of is in between these two, but at the asymptotes, you approach both a linear entropic spring, but then also a really linear enthalpic rod. A really important aspect is what you think about these polymers when they're under compression. Just like any stiff rod, when you compress it, it undergoes Euler buckling. So when you compress the rod, there's a buckling that occurs to these filaments under relatively low forces, so less than a piconewton. So now you can think of this as a very rigid spring under extension, but because of buckling, a very soft spring under compression. Okay, so that's just the polymer itself. So what today I'm going to cover is all the types of sort of soft materials and the states that you can get from actin filaments assembled. And then what happens when you start adding in local enzyme activity to make these materials have local mechanochemical activities to generate stresses. And how do you control these active stresses to direct what kind of morphogenetic or shape changes that you want in these materials? Okay, so you take these filaments and then you can polymerize them, and you care about two parameters. One is the length of your filament, and then one is the concentration, okay? You can control both of these by changing the polymerization states, but also just the total concentration of monomers. What most of the talk, one very, in a regime that's where we call the semi-dilute regime, there's when the mesh size, or the average distance between these filaments is on the scale of a micrometer or lower, you form what's known as a semi-dilute sort of entangled solution of actin filaments. And so here, because the actin filaments are so long compared to their average distance between it, they actually gives them a significant viscoelastic response, where it's all due to the steric entanglements of the polymers themselves. So that's this regime of very low dilute concentrations. As the concentration is increased, you pack these rods closer and closer together. And now if you make your filaments shorter and shorter, those rods can pack and rearrange to find their preferred packing density and arrangements. And so when you pack it sufficiently dense enough, so now we're close to like 500 micromolar, so orders of magnitude higher than the semi-dilute regime, you go to this transition to a nematic phase. And so on the right here, this is a fluorescence micrograph of actin filaments, of short actin filaments in nematic phase, where you can see the alignment and packing of the filaments so they form a liquid crystalline arrangement. At the transition between when there's complete disorder and high order, there's a transition between this isotropic nematic phase. At that regime, you get nucleation of droplets of the nematic phase. And what the structure of these nematic droplets you get at this transition are known as tactoids. And these are really droplets of liquid crystals that are nucleating and these really highly dense suspensions of actin filaments. Okay, so this is just actin filaments. Because of its rod shape, you can get enhanced rigidity of the solutions, even at a very semi-dilute concentration. There's a huge literature on that, probably from 20 years ago. And then as you increase the concentration, now you pack these closer together and you form a liquid crystal. Okay, and so then once you add cross-linkers to this, this prevents sliding between the filaments. So inside cells, there's cross-linking proteins that bind to the filaments and basically kinetically arrest these filaments and stop their mobility. So if you polymerize the actin filaments in the presence of this cross-linker, now you form a gel where actin filaments can be bundled and cross-linked together. And of course here, now in addition to those two parameters I just described, of the filament length and concentration, you also include the properties of the cross-linker and the cross-linker concentration. So, and the pictures here are two images of the types of networks and bundles of networks you can get of actin filament, cross-linked actin filament networks. And so these are gels, right? These are, the structure of these gels is entirely determined by how you assemble the system. It's not an equilibrated state. And so if we go through and now think about these two parameters of cross-link concentration and filament length in a regime where the filaments are overlapping, so in that regime where you would get a semi-dilute entangled regime of actin filaments alone, if you add cross-linkers to this, it turns it into a viscoelastic gel. And this gel, depending on how many cross-linkers you have, the elastic modulus can vary significantly of that gel. And in addition, you change from a gel that is stress weakens to one that stress stiffens as you change the concentration of the cross-linkers. So again, there's this whole gel state that's been well studied. Again, it's not, but if you're a student, there's a huge history of this. As you go down in filament length, but you still have cross-linkers, it was expected in the field that you would just like in the gels, with say colloidal gels, it's expected you to get clusters. So sort of kinetically trapped clusters of particles. But in this regime, you would not expect a space spanning network because your filaments are sufficiently short that there's no overlaps with them. So you would not get a full space spanning gel. So the first thing I want to just describe is something that Kim Weirich, when she was a postdoc in my lab, she just started her faculty position at Clemson, discovered when she actually went into this regime, which hadn't been studied before. And so when she reduced the filament lengths and started imaging over time in the presence of actin with a cross-linker called filament, what she saw were these little football shaped objects diffusing around in the microscope. So they're growing over time. I'll show you a series of images over time as well. And these, just to cut to the punchline, are liquid droplets of actin that's been assembled via attractive interaction of the cross-linkers. Okay, so these are little diffusing football-shaped liquid droplets of actin. And so how she formed them was from these really short filaments at a very low concentration, something that would be in the semi-dilute regime. She added this cross-linker called filament, and she formed these tactoidal droplets, just like what I introduced before. Just to emphasize the difference in what she discovered versus what had been done before, previously described, you expect these pneumatic droplets to form at 100-fold higher concentration just when there's crowding of an isotropic liquid surrounding. Here, there's really attractive interaction of the cross-linkers themselves. So if you look over time, you can see, and these are snapshots of the droplets over time, you can see that the size of the droplets increase, but their shape remains the same, right? The aspect ratio remains the same. And so the way these droplets are growing, you can catch these over time. They're growing by coalescence of the smaller droplets. So you can see this in the series of snapshots below. You have two small droplets that are diffusing around just by happenstance. They touch each other, and then they coalesce and relax back to a shape that's a larger drop of the same aspect ratio. Okay, she can frat these droplets and so bleach a portion of the actin filaments in the droplet, and after bleaching, the fluorescence recovers and becomes uniform throughout the droplet. So that reveals that there's internal mobility within the droplet. So there's rearrangements of the actin filaments. So we really thought these are actin droplets where the actin filaments are mobile inside. And I should say for those the cross-linkers that are forming these, a very important feature is that these cross-linkers have a pretty fast off rate. So they come on and off the filaments fairly quickly, which allows for these structural rearrangements, okay? So, so when we think about a pneumatic drop, and this is now just going back to the physics of liquid crystal droplets, the shape is determined by a balance of energies between the interfacial energy of the liquid crystal drop, but also the pneumatic energy of the liquid crystal pneumatogens. Right, so in the extreme example, if you only were dominated by the pneumatic energy, you want all the filaments to be aligned. If you're only dominated by an isotropic surface tension, you would get a spherical shape. And so this tactoidal football shape is really a competition between these two competing factors. And so we can tune those. And so this was work done by Kinjal Disbaswas and also another postdoc in the group, where you can really tune by changing the anisotropic interfacial tension with the isotropic interfacial tension, the shape of the droplet, to being from completely spherical and to being more elongated. So Kim did this when she was studying these the cross-linked filament by just increasing the concentration of the cross-linker. And as you can see by looking at droplets of a similar size, you can really change the aspect ratio of these droplets by just changing the concentration. More recently, Kim and a graduate student, Danny Sept, also found you can get these actin droplets by mixing the actin polymers, making sort of a mixture between the actin polymers and adding them into a disordered protein that undergoes a liquid-liquid phase separation. And so this protein, when it undergoes a liquid-liquid phase separation, forms a spherical drop. If you sort of dope in more actin filaments, you can induce a pneumatic energy to that droplet to form a tactoid drop. So this is a case where there's no actin polymers on the left-hand side. That's this lat, it's a shortcut for just a drug that prevents actin polymerization. But as you increase the length of the actin filaments, you can see that you can form these football drops. So really, this is a way for this biopolymer in these composite droplets to induce a pneumatic architecture to an otherwise disordered droplet. I've asked, I mean, as probably many of you know, this liquid-liquid phase separation field is very large. There's people studying liquid phases of RNA and DNA. To the best of my knowledge, that other biopolymers that have been studied with this have not seen this liquid crystalline architecture. As you see later in the talk, I would be super excited if anybody had any evidence that these other types of biopolymers could really induce this pneumatic organization, because it's a way to provide internal structure to this droplet. Okay, so really what Kim found was that there's this whole new range of space here, and she mapped out this state space of where we originally thought there were just these viscoelastic networks, these kinetically trapped viscoelastic gels that are formed by cross-linked actin. But as you reduce the filament length, you can get to a liquid phase where the structures are equilibrated. It is an equilibrated shape. And you can, if you go down and cross-link, this is a cross-link dependent phenomenon. If you go down to low enough cross-linkers, you don't get this attractive interaction. So what happens as you transition between these droplets of actin and these viscoelastic networks? So I'm going to go down this where these blue circles are. I'm going to show you data what happens right around this transition. Well, there, when you form it, you form these non-equilibrated threads of actin, of cross-linked actin. So you get these long bundles of actin that are just sort of like formed because they've assembled quickly. But then over time, the equilibration time is increasing. So it takes them a long time to recover their shape, and they are contracting. So this is a case, remember, in this liquid state, surface tension is actually dominating the shape of the system. And this is an example where surface tension is actually driving the contraction of these bundles because it wants to equilibrate to this tactoidal shape. And so we can characterize the surface tension that's required to contract the bundles. You can see that a few of them actually form tactoids by the end of the experiment. If you look at the movie, there's just a few tactoids at the top. But many of them sort of get stuck. The equilibration time is way too long. And if you also look closely with an eye for people who like threads of polymers, you can see evidence of Rayleigh plateau instabilities in these as well. So these are really liquid polymer threads where surface tension is driving shape changes and instability. Okay, so this is where, that's at this transition, right? Where you can go from being a liquid droplet to something where the relaxation time, stress relaxation time is increasing, but it's still surface dominated. Those are those blue circles. And into these kinetically trapped gels that, as far as we know, don't rearrange with time. So the relaxation time is so far exceeds anything we can measure by experiment that you really have to apply external forces. KT is not enough to rearrange them. So one question I posed at the beginning was really, how do you build a soft material that when you apply forces to it, it can change its structure in a way to adapt to mechanical forces at any range of length scale? And so one thing we're thinking about is where in this phase space do we want to build something that is adaptable? And so I hypothesize that it's really on this red line here. where it's at this transition of a stress relaxation time where KT might not be sufficient for stress relaxation on reasonable timescales. But if you add other sources of activities, you are able to drive those rearrangements. And in one of these transitions on the horizontal red line, previous postdoc Sayantan Majumdar found that networks really sort of just over the precipice of being from stress weakening to stress stiffening showed a mechanical adaptation. So these are gels where you can train them to stiffen in a direction where you've applied mechanical force to it in a reversible way. So one thing we're thinking about now is how do you think about how these soft gels can, what are the requirements for rearrangements in the soft gels in applications to applied force. Okay, so that was sort of this is now the state of what we know about phases. And now I want to ask what happens when we take external activities and add them to either of these states, a viscoelastic gel or a liquid. I'm going, for this talk, because it's more recent work, I'm going to focus on what happens when we add it to liquids, but I'm going to summarize what we found with gels as well, okay? Okay, so let's just summarize, let's just go through what the activities we're talking about. So proteins have enzymatic activity, or they can have enzymatic activity, and when they're mechanoenzymes, we call them a molecular motor. So one type of mechanoenzyme is just the actin polymer itself from its polymerization dynamics. So when you are growing an actin filament and through the asymmetry that is created from ATP hydrolysis, you can generate forces through growth of the filament. And so this is an example of a force velocity curve of an actin filament that's growing up against a barrier, okay? So that's one kind of molecular motor. It generates a pressure against a wall. The actin filament as a motor can also enhance stress relaxation. And I won't have time to go through this today, but by the process of disassembling the actin filament is a way to use activity to enhance the dissipation of mechanical stresses in the system. Okay, so that's one other activity. The other activity, of course, is if you add molecular motors that can slide actin filaments parallel to each other. So this is a cartoon schematic of myosin 2, which is a motor that's found in your muscles and also all non-muscle cells that can generate another polymer that then can slide two actin filaments relative to each other. So these polymers of myosin 2 motors also generate forces through relative sliding of actin filaments. Okay. So I want to emphasize here that the activity that's happening with the myosin 2 is a relative sliding, right? It's not generating a force per se. It requires a local geometry and material to generate stresses. So probably the best example of force generation from relative sliding is that of sarcomeric muscle. So in a sarcomere of muscle, this is an electron micrograph of a sarcomere. You have polymers of myosin 2 motors, actin filaments that are arranged with the proper polarity to the right and left of it. And then when the muscle is turned on, the motors walk along the actin filaments, increasing the overlap between the actin and myosin, leading to a local contraction. So this is where in this geometry, relative sliding is leading to an increase, a local contractile force. In lots of non-muscle cells, and basically all non-muscle cells, actin and myosin, shown here in an electron micrograph here with myosin in yellow, is completely disordered, right? So there's no sarcomeric organization. And here it's less clear what happens when you have two filaments sliding past each other, what kind of forces are generated, okay? So for a while, we were really interested in this. And this is a summary of the state space that previous graduate student Samantha Stam found a few years ago, where when you have this sliding, depending on how much connections there are in your network, so how many crosslinks per filament you have, that's the connectivity, and how stiff your filaments are, you can transition between different types of local stresses from the same actin-myosin sliding, okay? So the first one I want to just go through briefly is this contraction via buckling. So this is what is shown in this movie to the right, where only a sparse number of filaments are labeled, and you don't see the motors. But as soon as you add the myosin motors, you can see the filaments themselves are buckled and compressed. And so in here, what we found was that the contraction that's occurring is occurring because there's an asymmetric response that, remember that mechanical asymmetry of the filament to stretching versus compression? Well, because of that buckling of the filaments, this contraction really happens as a ratchet because of that, that asymmetric mechanical response of the filaments themselves. And because the filaments buckle, you get a local isotropic contraction. If you increase the rigidity of your actin filaments to the point where your myosin stresses cannot bend or deform, or the Euler buckling of that bundle gets too high, you can also get a contraction, but now this is contracting because there's a ratcheted sliding, which is basically if you have crosslinkers that are coming on and off, your motors generate some increased overlap, and at the point when you have increased overlap, your filaments, your crosslinks accumulate on that increased overlap, and at some point there's too many crosslinks for the motors to overcome, so you can never get to the extension phase. When you decrease your connectivity, now you could overcome that and actually generate extensile force. And in this case, when you have low connectivity but a high filament stiffness, now this sliding leads to a net extensile forces in the system, okay? So Sam, when she was a graduate student, measured the looked at the strain tensor of the networks while they were contracting or extending, and showed that there was this net extensile or contractile force. As an aside, for those of you who, and sort of as a, a foreshadowing, so people who know Dan Needleman's work on microtubules and motor proteins, right? Microtubules are more rigid than actin filaments. So when Dan Needleman described contraction, it was all in this contraction via ratcheted sliding, right? Zvonimir Dojic, and as I will show, pneumatics are formed in this really low connectivity. So that's all extensile forces. But this is one of my favorite things to tell biologists, is that you can have the same two molecules, and depending on your local lengths and mechanical environment, you can really generate different kinds of forces, right? That's why you really need to know soft matter if you want to understand morphogenesis. Okay, so as I was saying, today I'm going to emphasize on this extensile regime because, well, it's newer work, and it's newer. Yeah, that's probably why I'm focusing on it. And I'll try to focus on not on the stuff that, because I know many people have heard talks from the active pneumatic field, so I'm not going to go too much into the weeds here. But to make a long story short, we were really unclear whether we could form active pneumatics with the softer actin filaments and myosin 2 motors, as say could be done with the microtubule kinesin system. Nitin Kumar was a postdoc in the lab, and he showed a few years ago that we could. So using the same exact system where you, over a cover slip, you put an oil layer and then a surfactant layer and lay on top of that the biopolymers, you can form a pneumatic phase of the actin filaments and then add in myosin 2 motors and make it an extensile pneumatic. And the way we know it's extensile pneumatic is because of the way the +1/2 defects are forming, okay? And I should say that, like, when Nitin started working on this, it was shocking to me because it was the first time in my life that the theory had already been done. So we were able to really compare against, I mean, the physics is in some sense really boring because it's already so well known. Sorry for any active pneumatic people out there. But we were immediately able to team up with Wanda Pablo's group who had built a full hydrodynamic simulation of the active pneumatics to compare our experimental data to the simulation data. And for anybody who likes comparing data, it's it's a great system to do that with because the theory is well understood and the data is easy to collect. So, so we were able to like look at how the level of active stresses changes the effective Bend modulus of the liquid crystal. So what we found was that the shape of the +1/2 defects, as you tune up the activity in the system, it actually distorts the defects to make it look like the Bend modulus is actually softer than it is. So that's one effective activity. So you could sort of rethink the whole active system as something with just a softer Bend modulus. And also because it's an active fluid, now we can also think about organization of the topological defects, the plus and minus 1/2 defects in the system, and to think about the sort of the larger scale rearrangements, but also the local effects of activity. So this is all in this paper from Roy Zhang, a postdoc in Wanda Pablo's group, and Nitin from a few years ago. What I want to discuss is our newer efforts to try to understand how to control the activity in these systems. And so we really are thinking about two things now is so far these active biological systems have really taken the types of activities that are naturally found, meaning that the motors with a known force velocity behavior, and not really thinking about what the consequences of changing motor mechanochemistry are, but also how do we spatially and temporally control the active stresses. So to do this, we've been, over the past few years, we've started collaborating with Zev Bryant, who's a molecular engineer at Stanford, who really has designed different ways of engineering motors, and his postdoc, Paul. And so what I wanted to show you some of our data on what happens with a tool that they developed called gear shifting motors. And in these gear shifting motors, so here's their schematic of a molecular motor doing 2 steps. So to the left is the first motor step, and then after it takes 1 step, it's moved. And here you see the feet where it binds to the actin filament in black. The actin filament is red. And basically what happens when you shine light is you extend this green protein portion of the protein. So it increases the step length of the molecular motor without affecting the chemistry, the chemical cycle or the kinetics of the motor domain itself. So you're increasing the step size without changing the rates of these transitions. Okay, so what this does is change the velocity of the motor. And so here is data from their lab showing what happens when you turn on blue light. The velocity of the motor now can be increased by over a factor of 100 simply by shining blue light on the system, okay? So we can put these motors in our pneumatics, and now the pneumatics serve as a really great toolbox because their physics is so well understood to understand what how these local mechanoenzymes are affecting sort of the resultant emergent active mechanics. So this is an example of a pneumatic where we have activated using blue light those motors, and you can see that there is a lot more motion and a lot more defects that exist in this activated region than say outside the region that we illuminated. We can characterize the number of those defects both inside and outside the border. We can look at the trajectories of those defects, and you can see that most of those defects get constrained in this region, while only a few leave. And if we zoom in at the bottom here, one of these defects that's approaching this boundary, you can see it goes in perpendicular to the boundary, and as it's reaching this boundary, it gets deflected and then turns away from it. So this is an experimental system where we can start to look at what happens at gradients of activity in the system to understand how there can be control and how to control the motions in the active systems. And so one of the first examples of just trying to control the defect motion is that Stephen created this half or quarter annulus where he only shined light in this region, and then he sort of captured one defect, and he was able to tune down the activity so he didn't create too much new defect nucleation, but he was able to guide the defect trajectory around and to shape it. And so here's this quantification of showing how he's able to use light to direct the flows and motion of the defect in the system, rather than just getting a chaotic turbulent system. Yeah, so David just asked, so all these systems have ATP. One of the reasons is the motion slows down at the end is from ATP depletion. So it does deplete its ATP over very long times, but we don't continuously feed it ATP. We just turn on and turn off light. The ATP is always around. Okay. And so this is sort of inspired and a lot of theory work and modeling work from both Juan de Pablo's lab and now more recently Vincenzo Vitelli's group to understand how you build spatial active materials with spatial constraints to it. So some of the things they've been looking at is both what are the effects of having the gradients between the active and passive system as a way to pattern and control the active turbulence in these pneumatics. What are the geometric constraints and the activity gradients required to nucleate new defects? So how do you act, what's the, sort of at the microscopic level, how do you create, what's the energy cost to create new topological defects in the system? And also, what are the local elastics and stresses that are required to direct defect flows? So they have the simulations worked out so that they can really explore these a lot, that we can then go back into the experiments to do a much fewer set of experiments. So it's really this nice feedback between simulations that then are testable by our experimental system. Okay, so for the very last part, and this shouldn't take more than 5 minutes, I want to go back to the droplet phase. And this is sort of fun icon candy. So this is again actomyosin in a liquid droplet phase instead of the 2D active pneumatic, but it's still a pneumatic, right? So now Kim Weirick asked what happened when you add these myosin 2 motors to these little pneumatic droplets. And what you see from these two movies is that the myosin shown in white here migrates towards the center of the droplets and over longer time clusters to form a big clump and then eventually bisects the droplet in half, okay? So this motor is able to find the center of the droplet and divide it in a process very reminiscent of cell division, right? These are two very fundamental processes that you see in cell division. So the process of centering in the droplet, Kim found was independent of motor activity. It actually did not require any active stresses. And so here, this is a graph of just motors that don't have a lot of enzymatic activity. All of them find the center of the droplet. None of them divide the droplet. So you require enzymatic activity for division, but not for centering. And the way we thought about this is the fact that you have this pneumatic droplet over here on the right in this cartoon, and now you're placing a rod. So the myosin filament itself is a rod with a length on the order of half a micron. And so now you're placing a larger rod in this pneumatic droplet. And because there's more splay at the poles, the ends of this droplet, this rod migrates to the place of minimal splay. So if you calculate the free energy as a function of distance from the midplane, the energy is going to be minimal at the center of the droplet, perfectly at the center. And this, to verify this, to verify this idea that you have higher potential energy if you start farther away from the center, Kim measured the initial speed at which the motor migrates towards the center as a function of distance from the midplane and found that it went up, okay? So the process of finding the middle of this pneumatic droplet is just the motor, which is a rigid rod, finding its lowest free energy state. So the process of bisection, as I indicated before, requires a high degree of motor activity. So you need to have a high amount of enzymatic activity for these for the motors to actually divide the droplet into two. Okay. Kim measured these droplets always are bisected into two equal daughter droplets. So the length of these two daughter droplets are exactly the same. They didn't end up showing any images of this, but the droplets actually can separate from each other as well, leaving like a droplet with one pole on it as well. And so here we got a little bit more hand wavy and where it requires more experiments. But the picture we have is that, you know, this initial step of finding the center is really just a minimization of the free energy. And then there's this magical step at in this cartoon of the third to fourth image over, of where all of a sudden these rods become a clump. I'll just call it technically a clump. And I don't know what else to say about this. Actin-myosin forms these like, tends to form clumps. If we form a 2D dematic, it does clump together and form these star-shaped patterns where now bundles and filaments are pinned in a star arrangement. I would love to have a soft matter system to be able to control this more precisely, but that's about all we could do with the native proteins. So there's this process of going from individual filaments to a clump that we think then changes, again, the preferred anchoring geometry of the myosin to the actin filament that then makes it prefer to divide into two daughter tactoids. And that's our thought about how bisection occurs. Okay, so, but it's a really fun system because basically what we've done is reconstituted the process of center finding and division now with 4 proteins. And that's all you would need. And so I would, again, if anybody likes to work with other kinds of liquid droplets or DNA-based materials, I feel like there's a wealth of probably very difficult experiments that can be done to test ideas of how you could build self-centering systems or division systems in other types of structured droplets. So just the last thing I want to say is like this last story was really, you know, I think what I learned with the light mediated control of the actin nematics is that is a great way to sort of interrogate how active stresses affect the system, but it's definitely not how biology does it, right? Biology uses these local feedbacks between activities and either geometry or force sensitive repair, remodeling to really drive morphogenic changes. So in the future, that's really where we're interested in going, is how do you use your local structure to either turn up or turn down your activity, or how do you use force sensing and force feedback to drive morphogenic changes? And that's really what we see happening in cells, and we'd like to be able to recapitulate it in our soft matter systems. And with that, I'd just like to thank, of course, my lab. This is a picture from early this year. In red are all the students, the current and previous students that have worked on these types of projects in the lab, as well as our collaborators. And also our MRSEC is hosting a Rising SARS in Soft and Biological Matter on October 8th and 9th. If you're looking for a postdoc or a great faculty candidate, come to this and meet all the people, the 15 excellent people we've selected. I see Sophie Marbach on this talk, and she's one of the people we selected. And also our physics department is hiring at the assistant professor level. So if you know anybody, and it's in all fields. So if you know anybody who'd be a good candidate, please encourage them to apply. All right, and thank you. That's great.
Yay!
It's so much better. It's so much better when it's real audio.
Great. So Margaret, I'm since no one is organizing this, so I'm just going to dive in.
Okay, should I stop my screen sharing?
So you said something pretty like provocative, right? That that this system models center finding in cell division. So, but there are differences, right? Because how is this related to like the spindle microtubules centering the centromeres of chromosomes? Is there a relation?
Yeah, so there's no, I mean well, okay, I guess I would say two things. Yeah, this is only showing it's a way of doing it with that's probably not that robust, right? So it's not saying that this is how the mitotic spindle works. Although Dan Needleman and Mike Shelley, right, have this model of the mitotic spindle as a liquid crystal droplet, right? And so this, I would say, would be evidence that you could actually, before, when they proposed that model, to the best of my knowledge, nobody actually showed liquid crystal droplets experimentally like the ones they were describing. So to me, when we were able to form liquid crystal droplets in vitro, I was really excited because that meant that the idea of a mitotic spindle as a liquid droplet. I could imagine how you form a liquid crystal droplet that way. But yeah, this doesn't have any error correction mechanisms in it like a mitotic spindle would. So yeah, you're right. It's not shedding light on the biology, I don't think. It's showing a sufficiency. I would love, so my fantasy is you would build like a phase separated liquid drop out of like RNA, right? And then this is a way you can replicate your RNA, right? As like a primitive cell, right? That's where I would love to like see if this could work. I don't think it's going to shed light on a eukaryotic spindle.
Can I continue on that same topic? So your description was that initially your myosins diffuse towards the center because they want to go to the minimal free energy. That I buy. Then they start building for themselves their maximum of free energy because they build a place of high splay.
Yeah.
So what is the switch? What forces them to do other things? What happens? Why they start doing something new?
Yeah, I honestly don't know. I don't think they're doing anything new. I think
you said they do something diametrically opposite.
So we see a lot in so we see this in a lot of the actin-myosin reconstituted systems where that over time as actin-myosin is contracting, the aggregates they form are disorganized, but then also have this they tend to form arrangements where the filaments are splayed out. But you're right, this is about this that requires enzymatic activity. It's not just motor binding to actin filaments. As far as I know, that's not seen in other in vitro systems. We've seen it in a lot of the actin-myosin assemblies. And these clusters are seen in cells too. I think I don't know, I'm not giving you a good answer.
Margaret, I think Sasha Bershatsky had this cool paper a few years ago when he showed a similar thing in actual cell. Alignment of this myosin bands, right? I think that's
well in the puncta. Yeah, I don't know.
No, they were assembling into stripes across the whole cell, and I think it's kind of perfect in vivo analog of what you're showing in vitro.
I have to look at that. I mean, I've seen the stripes along a bundle.
No, it was completely disordered actin myosin gel in the beginning.
Yeah.
And then the first stage of stress fiber assembly was this long bands of myosin with actin kind of.
Yeah, but the question was really like, why do you get to a point where you have like a preferred anchoring of being parallel to perpendicular to this spot? And I don't have a good like mechanistic way. It has been observed a lot. I don't know.
But I'm actually asking a simpler question. I think I'm asking a simpler question because I'm asking who gives the order? What is where do they receive a signal that they should stop diffusing?
Oh, okay. So all right. So again, this is not purely technical, but the signal I think comes from the concentration, right? It's really from how they're contracting and
concentration of myosins.
It's based on the myosin concentration and the distortions of the local actin. Yeah.
Right?
So
when they many of them come together to the middle concentration.
That's right.
Threshold.
That's right. And I mean, for people who like myosin too, I actually think it has something to do with like the tails interacting, the interaction of the myosin tails. But this is seen a lot. That kind of clumping is seen a lot in vitro systems, and I think it has purely to do with an effect of concentration, local actin and myosin concentration in their formation, and some. Yeah, cells relieve this by disassembling the myosin polymers before this happens. But we do see this in vitro quite a bit.
Thank you.
Any more questions from the crowd?
I'm happy to.
Yeah, if you,
Oh, okay.
go ahead.
So I think you mentioned it a little bit, Margaret, but do these tectoids, are they observed in vivo? Like
Yeah, so that was there. As far as I know, I have not seen them in vivo. I mean, I've asked around a lot.
okay, so the cells are working in a concentration range or whatever physical parameter range that makes them not in that regime.
Yeah, or not easy to regime. I mean, get the concentration regime that cells are working in. I think they sort of move fluidly between like the gel and the fluid phase, but when they get into the fluid phase, actin filaments disassemble. So you would never see a stable liquid phase. That's my, there's no reason why they shouldn't exist based on the concentrations in the filament lanes. I just don't think they're stabilized. Somebody, a developmental biologist told me once they thought they saw something like this. I mean, tectoids of amyloid fibrils have definitely been seen, right? So there's other examples of biopolymers that I've seen, but I think that those types of liquid crystal droplets of actin have definitely not been seen. It's like one of the, yeah.
Here? So if you deplete ATP, you'll still get the myosin going to the center, right?
Correct.
But it won't. But it then won't start dividing. Is that right?
They will not divide. Yeah.
And now, if you control it, if you just add more, will you see it, and will it stall, or will it? What will happen if you try and just control the rate at which this happens with ATP?
Ah, yeah, that's a great question. Kim wasn't able to do those experiments. Yeah. So, so you're asking about whether it would actually end up dividing when there was a high enough concentration of myosin?
Yeah, and also, the rate, will it stall? Does it need a certain activity? Does it need what?
Yeah, no, those are great questions, Paul. We have not done those experiments. I mean, so that's.
Okay, but they certainly will go in because of the diffusion. They'll still go in and order it and go
That's
to the
right,
center,
and the binding.
right?
Yeah, that's right. Yeah. Yeah, there was we did do some concentration range, but it was limited in terms of its ability to really explore. Glenn Hockey had a question.
Yes.
You said
Simulations show the effect without clumping.
I'm a little confused by his question.
Well, I'm guessing
I mean, in the model, in the theory that we put in the paper, there was the clumping process was just put in by hand. Yeah, the clump, I mean, there was no, yeah, that it was not, we didn't have a microscopic model of the simulation of the rearrangements. Oh, thanks for putting the link to the Rising Star program.
So I have a silly question, which is, if you put these droplets that become tectoids together, is there a way to make them interact in any way through coalescence or any kind of merging, so that you then sample the different geometries? Like,
yeah, so that's a good, yeah, so that's one way that Kim was able to really watch the migration process was during coalescence, because I mean, these were really technically challenging experiments. And what she was able to do, she had to, if she like blew on them the wrong way, which is like pipetting, everything just disintegrated because they're very fragile. And so what she was able to do was during a coalescence event, that's when she was able to get myosin that wasn't located at the center, because that process was really fast. So actually a lot of that data came from coalescence data.
Mm-hmm. And conversely, could you make the system such that there's surfactants or other stable lipids or some other stabilizing components that would make them stable?
Yeah, it would be great. I mean, I think if I were if I could convince a student to work on this and get funding for it, I'd probably work with these actins with this disordered protein fuse, because those are really easy, much easier to make. This disordered protein fuse, you can make composite networks out of them. I mean, the other thing that I think I would really just try to get rid of using the native myosin 2 proteins and work with other kinds of engineered constructs, because then you can do all these questions that people are asking about, changing like geometry and changing concentrations more precisely, and doing systematic changes. She was really limited with the native proteins that she was working with. Yeah, that would be cool. And the fuse one seemed a little bit more stable. So I don't know, there must be people working on adding surfactants to those liquid phase separated drops. I don't know. I don't follow that field as closely as many others do.
So Margaret,
I mean, I know that you can get, if you do liquid phase separation and you put in mixtures of surfactants, then you can go, depending on the surface tension ratio for the different surfactants, you can
That's
go
cool.
from having multiple emulsions to
Yeah, that's right.
having
That's right. There's like a pen doing that. Yeah,
segregated into compartments to ones that you can get different outcomes to how.
yeah, that would be, those would be super cool experiments to do. Yeah, that would be neat.
so actually I wanted to ask if the different morphologies, the different dynamic states, if they affect the ability of these things to be mechanosensors. Like, so if you have those sort of like force sensing proteins in the middle of these networks, does it matter which state you're in? I imagine it does.
Which state, which are the
Well,
two
like,
states?
like, for example, if you're tending to form an active pneumatic, or if you've showed us a whole phase diagram.
Yeah,
Each
yeah.
element of that, or state diagram, each element of that state diagram presumably has different ramifications for force sensing.
It should, right? Yeah, no, it should. I mean, yeah, so that would be, I mean, we have not explored that. I mean, the force buildup in the liquid phase is very minimal, right? It's not,
Right.
Yeah, so I would think that you would really need to be in the gel phase to do any sizable force sensing, but there should be force sensors of all different, levels being able to provide that.
Yeah, but because there's the isotropic gel, there's the pneumatic gel, and they probably are used for different things.
Absolutely, yeah. So like, I mean, Dave Kovar, my colleague, there's different actin cross-linkers that will only bind to filaments when they're aligned, right? And will not bind to them when they're disordered, right? So you would think that all these different ways to build or have local feedbacks between the geometry and the stresses could be used. I don't know of a set of experiments that have really explored that, but that would be really cool.
Margaret, another one?
Yeah.
With your super activated light sensitive modification, where you got a factor of 4,000 in velocity, is that what you said?
That's what I mean. It wasn't that's really, so the motor was basically off and then it got turned on in that data.
Okay, that sounds better.
Yeah.
And it consumes much more ATP and faster, is that right?
That's correct. When it's going, well, no, the enzymatic activity, the rate should be the same. It's just the stepping is not, he can change the step size by changing that angle between the heads. So so he can actually modify whether it's step or not.
So how much faster is it than regular myosin when it's not sleeping?
Oh, so he can make motors. It's really cool. He can make motors that are like 100 times faster than myosin 2 and 100 times slower. I mean, he can really do the full range. The light sensitive ones are more limited, but and those when the ones that are faster, that's both because the step size is different, but also the enzymatic rates are also different, and those consume ATP at a much faster rate.
So what happens if you use those, in the division of the tactoids?
Well, that would be super cool. I would love to even, my graduate student who I should just, who I asked for data for this talk, we were just looking at them in the active pneumatic, and what's super cool is that even in the pneumatic, just change the enzymatic rate of the myosin, gives you totally different defect density and turbulence. Like, it's not scalable, right? You change the local mechanoenzyme and it doesn't give you the same active pneumatic. You can't parameterize them the same way as other types of mechanoenzymes, which I think is super cool, because it's really telling you that there's like a local effect of force generation that we don't understand. But in the tactoid, that would be a totally different question. Yeah, we have no idea in the tactoid either. So the tactoid, I think the key challenge in the tactoid is building the backbone of the motor dimers, because then you have to build a long rod. So the experiment I would love to do in the tactoids is make the backbone with different lengths and different flexibilities to see what geometric effects of this motor dimer are necessary for the centering and division process, even to understand that clustering better, which we don't understand.
And then do laser ablation.
I know, it's just, I know. This is getting the realm. This, you can just plug your ears, but these experiments are very hard.
Yeah. Cool. I was wondering if any PhD student or postdoc wants to ask a question just for the fun of it. I know everyone is looking a lot like black boxes, but I know there's a lot of diversity in the pool of listeners. So I'm just wondering that we haven't left anyone out in the discussion. No? Okay. Making sure, giving time, giving space. Go for it, David.
Well, actually, what I was going to say is what we might do at this point is maybe unmute and thank Margaret properly, and then turn off the recording and just keep talking.
Okay, sounds great.
Can we do that?
Yeah, let's do.
Thank you.
All right, that was great.